it's going to be work that I did with Michael Desai, Mark Feldman, and Dan Fisher, and that I'm doing some continuing related work with, where I'm kind of tagging along with Joanna Maisel and Meredith Trotter, some other stuff with Yoav Ram, and then a lot of other people have worked on something like this, including a lot of people in this room who I'm not mentioning. I'm just going to mention Nick because people may not know that actually he has worked on this. He may even deny it at points. And why would he deny it? Because the problem is something a little bit makes biologists a little bit unhappy sometimes, because it's the evolution of irreducible complexity. If you have some kind of adaptation that needs multiple mutations to do anything, how can evolution ever find this? And then it has also some less loaded names like Fitness Valley or Fitness Plateau that I'll probably use instead. And I'm going to say that you need K mutations to get it. Okay, so why should we care? Actually, despite its kind of bad reputation, we are pretty sure that this is a real thing that happens in biology. You can imagine if you've got like a signaling protein and a receptor protein, and they need to stick together, and there's a better way that they could bind, but they need to change in concert. All right, that should be an example of this kind of adaptation. The kind of most clear-cut case is cancer. So the cells in your body, okay, they can see cancer as an adaptation on their part, at least in the short term, because they get to take over and kill you. But okay, you don't like that at all. So you've made it. We've evolved to make it very hard for cells to turn cancerous. And in particular, there are a lot of genes that will kill them if they start dividing out of control. And since you have 2 copies of every gene, you would need to at least inactivate. A cell would need to at least inactivate both those copies to turn cancer. So that's at least a K = 2 adaptation. And then kind of more generally in this audience, we're interested in, can evolution get stuck? Should evolution proceed by single substitutions or by selection on interesting combinations? And a couple people have been saying that it's interesting to think about because there's so many possible genotypes that are, say, 3 mutations away from your current genotype. Maybe one of those is something really cool that we'd be interested in. Okay, but there's 2 problems with this that make people a little skeptical about it. The population has to first produce this combination of different mutations, and then it has to fix it, by which I mean everyone needs to acquire it in the population. So I'm just going to work backwards here, start with the second problem. And now we're going to kind of forget about where this combination of mutations came from, but just say it's in the population. Under what circumstances can it spread, starting from some very low frequency or very low number? Okay, so the kind of classic challenge that has to overcome is to avoid being broken up by recombination. The adaptation can't get split up by recombination. So say it's at some low frequency. Is this thing really not working? All right, I'll use my hand. Okay, so say it's at some low frequency, and it's increasing because it has a selective advantage, but it's getting broken up by recombination. You have a competition between those effects. You also have recombination feeding in new copies from the other mutations that are present in the population. And then you also have stochastic effects over there. So first thing to notice, if the recombination rate among the different involved alleles is smaller than the selective advantage of this adaptation altogether, well, then they don't even need this influx from recombination. They can just spread. They don't even need to worry about getting broken up. On the other hand, if the recombination rate is much larger, then you need this influx to almost exactly balance the loss due to recombination, and selection is just going to be a little added term on that, a perturbation. And I just wrote this as some function of the mutant allele frequencies. In the absolute simplest case, it's just the product of, where there's just 2 mutations necessary. It's just the product of their 2 frequencies. And so that's kind of the justification for what you saw in the last talk, where you assume that it's kind of pinned the distribution of genotype frequencies is pinned very close to the product distribution by recombination. Okay, so what does this mean? This kind of competition between selection versus recombination? Say we have some rare combination given a 1% advantage that shows up in some very low numbers in a population. Does it need to worry about being broken up? So 1% selective advantage corresponds to recombination rate, 1%. In flies or people, that's 1 million bases on the chromosome. But the gene densities are different, so it's 100 genes in flies, 10 genes in humans. If it's within that, if it's within those number of genes, then it doesn't need to worry about it. Otherwise, it does. For yeast, so for anything that is not an obligate sexual organism, like a lot of animals are, it's very hard to measure recombination rates. And it's not entirely clear that what we measure, well, it is clear that what we measure when we say we're measuring recombination is not the relevant quantity here. But does anyone want to object if I say that yeast, we could use mutations anywhere on the whole genome? Probably 2 random genes reassorted at a lower frequency than that. Yeah, I think that's probably likely. HIV is actually one of the few organisms where we do have some kind of reasonable estimate of the recombination rate when it's just evolving within a host, not at a population level. And actually there it is just about 1%. So that only gets 1 question mark because it does seem more reasonable to say it's the whole genome, anywhere they can evolve together. With bacteria, they don't evolve by crossovers. They evolve by, they don't recombine by crossovers. They recombine by shuffling genes, taking just one gene or small stretch. So that means not only could this combination involve genes from anywhere in the genome, it could involve basically all the genes. That's not realistic because you could never change all of them at once. But given that you did, the whole genome could basically propagate together with an advantage of 1% without being broken up. Yeah.
So it can also be whole genome if the population was strongly subdivided, or sexual but selfing, for example.
Yeah. So this is some other issues that with HIV you could kind of get into that. E. coli too, strongly subdivided population. So I was saying with HIV we know the recombination rate within a host, but 2 people with HIV, their strains are only very rarely recombining, only when they co-infect someone else. So then it's a little different, but it gets a bit technical. I mean, it is something to worry about because it would still need to spread within a local region. Yeah. And then for cancer, it's definitely whole genome because there is no recombination. There's mitotic recombination, but that doesn't act like this shuffling genes. It acts like mutation. Okay, so actually this selection versus recombination thing is not a problem for most of the tree of life, or a lot of genes in even animals. What is a bit more of a problem is just stochasticity. So fortunately, we already know all about branching processes. So I can just say that 1% advantage means that if I start with 1 copy of the one individual who's got that adaptive combination, there's a 99% chance, order 9, there's a very high chance that it's just going to die stochastically. And so that means we need, to produce this, to get this adaptation to fix the population, we need to produce it, 100 times. Order 1 over s times before one of them will be successful. Okay, so that's a bigger problem, and that's why the real issue, that's why this is a very serious problem. How can the population ever even find an adaptation that needs a lot of mutations? All right. So the answer is if you need a combination of enough mutations, it can never do it, right? It can never find one particular combination where you need to change every single gene at once in some complicated way. Okay, but how about for some limited level of complexity? So like I said, this is kind of practically important for a lot of things we care about. The number of potential genotypes is growing exponentially with K, like I said. So even being able to do some low K means that you get to explore way more genotypes. and potentially find something cool. Okay, and we just want to get some rough sense of how this question might depend on what kind of population we're looking at, because different populations are going to vary widely in their ability to find complexity. Okay, so just absolute simplest model, because this is going to get a bit complicated. Say that you just need 2 mutations that need to work together to get a benefit. When they do come together, they give you an advantage S. Either one of them has some small cost delta, could be 0, and they each occur at a rate mu. We have just constant population size, big N. And for now, let's say it's asexual, so we don't have to remember one more parameter. And I'm going to focus for now on the mean time to adapt, which is not relevant for most kinds of cancer. But if you want to know about what's relevant for cancer, you can ask me later. And okay, so there's a lot of parameters here, so don't hesitate to ask me to say again what one is. Okay, and already it gets really complicated. So don't try and understand this here. This is just a picture showing if I vary just 2 of the parameters, the population size and how bad those single mutants are for you when they're individually present, the different possible kind of dynamical regimes that can describe evolution. Okay, so too complicated. Let's just simplify down even further, focus on the fitness plateau case, where we're going to assume that the selection against the single mutants is weak enough so that we don't need to worry about it too much. All right, so first guess. Let's say we write down the frequency for the frequency of double mutants as a function of time. Got a nice differential equation there. They're produced by mutation starting from 0 frequency, increased at rate s, and you can solve it. Find when x2 is of order 1. Very simple. There's a bit of a problem here, though. It doesn't really depend very much on the mutation rate. It kind of makes you worry. And obviously we cheated because we let this selection term kick in even when x2 was less than 1 over n. Like when there was, a tenth of an individual who had the adaptation in the population. Okay, so that doesn't really make sense. So we're going to need to, this would only be valid if there's tons of double mutants produced in the time it takes for selection to start increasing them. Then, in this case, we get a big influx of mutants, then selection acts, and we don't have any worries about selecting on nothing. But if we go to kind of general K, more than 2, this is a very stringent condition. We need the necessary population size for this to be accurate is growing exponentially with K, and with it looks even worse. It's growing as K factorial. But remember, we're talking about moderate K, so actually here the constants are more.
U has to be bigger than S times K,
basically. Bigger than S times K? Oh yeah, the K, yeah. You need very strong mutation. You need mutation to be the dominant force in the population, stronger than selection. And when that's true, you can see that the model is kind of broken, because how are the mutations knowing where to go? Implicitly, I've assumed that we can neglect all the mutations that aren't leading anywhere useful. Actually, when mutation is strong compared to selection, you're getting the whole population spread out over genotype space. So the model breaks down there too. So this is problematic. Okay, but we got the answer. We got one of these regimes now. The very big population case done. Okay, 2 more to go. Second guess. All right, the problem was that we were selecting on nothing. We need to take the 0-1 boundary between for the double mutant seriously. But okay, let's make another approximation. If this is the problem, getting that first double mutant, let's say that that's basically the only problem. All we need to do is get that first double mutant, and then the first successful double mutant, right? It'll take 1 over s of them, and then once we do that, we're good. Selection will fix it quickly, no problem. Okay, so we say, all right, what's the frequency of single mutants now? No selection on them, so we don't need to worry about selecting on 0. It's just given by the mutation rate times time. They're coming up in frequency. We're not in a steady state here. We're out of equilibrium. Each one is producing double mutants at a rate mu. Can find exactly when you get 1 over s double mutants produced. All right, and there we go. We have a new answer for our time. Okay, and now it does depend on the mutation rate. That makes more sense. It's a little worrying though, because it only depends on the mutation rate to the first power. Like there was no need for a second mutation at all. And so that's kind of telling you that one of these mutations is not rate limiting at all in this case. And the natural worry is that maybe we cheated by ignoring the stochasticity in the single mutants.
Stochasticity, you mean the chance that it's going to die?
So here, if I want, I wrote down a deterministic expression for the single mutants, right? So there's a chance that they die, or maybe that they get lucky, right? This is just the influx due to mutations. Maybe they're spreading, right? So basically what we need is this mean value to be very big compared to the fluctuations, the stochastic fluctuations in the number of single mutants. And in particular, we need it to be large at the time where that successful double mutant is being produced. Maybe at very early times or very late times the picture is different, but we don't really care. We just want to get this one time scale right. Okay, so let's check by treating all the mutants stochastically, singles and doubles, and see if we get a different picture. Okay, so now the branching process stuff is gonna really come in handy. Here's a picture of a stochastic realization of a single mutant lineage. Start with one individual. This is a very unusual one that happened to get lucky and reproduce lots of times, but eventually it will go extinct with probability 1. And I've just left some 1 over deltas on here, even though we were ignoring it, just to give you some sense that we shouldn't be hitting those kind of scales, otherwise our approximations break down. And the key...
Remind us what is delta?
Oh, delta is the cost of the single mutants, right? Which we're assuming is small enough, and this picture is going to just kind of show you what small enough means. So the key thing that we care about this stochastic lineage here is the area under the curve, the total number of progeny of the branching process, because each one of those progeny had a chance to acquire a mutation, become a double mutant. So the total probability of success of this lineage conditioned on this realization is just the number of double mutants it produces times the probability of fixation of each of those double mutants, which we said before was S. So it's just going to be the area times the mutation rate times S. All right, so we need to know the statistics for this area, the distribution of this area. And all right, we know it's a branching process. It's all very simple. In this case, it's, I'm actually saying it's a near critical branching process. So that means it's going to die out, but like we heard this morning, it's going to take a long time to do that. It takes a long time for the extinction probability to approach 1. In particular, there's a probability 1 over t that it survives for some t generations, and given that it does, it has order the same number descendants at that time. These are just facts about branching processes. They're pretty easy to derive, though. Okay, so t generations, length t, height t, means area t squared. So put this in. The probability that you have area at least a is probability that you live for time root a, because then you also have root a individuals, area a. And so the probability that you get area at least a goes like 1 over the square root of a. We have a very long-tailed distribution here, only falling off like 1 over, the cumulative distribution is only falling off like 1 over the square root. What that means is, let's say I had some population, I'm watching it for a long time, and what I really care about in that population is the total number of single mutants that have ever existed up to this time, because that's the number of opportunities to acquire a double mutant that the population had. What this says is, I can just look at the largest lineage of single mutants that will be a good approximation. All those other thousands of lineages will contribute a negligible amount to the total number of single mutants. In other words, the most likely path for a single mutant to be successful is just, the most likely path for the population to be successful is just to have one enormously lucky single mutant that persists for a very long time, generates tons of double mutants, enough so that it easily produces 1 over S and crosses the valley, conditional on the time it survived. And you can, straightforward, you need the area to be 1 over mu S, so you need the time to be 1 over root mu S. This occurs with probability mu S, get an expression for the total waiting time. And you can see it here. This is a zoomed in picture on an actual, on a simulation, where we've got blue and yellow single mutants. When they come to, if you get both, you have green, which is obviously better. And you see, you get one big lineage, and that's when you cross the valley. And the full population size is way off the screen. This is some tiny fraction, but huge absolute number. Yeah, exactly. So yeah, and then this is just even more zoomed in, and you can see it actually does have the right dimensions that it should. Okay, so that now we have we filled in all 3 of these regimes, and in the process, I didn't explain it to you, but we actually found the boundary where you can distinguish, where you need to take the stochasticity of the single mutants seriously or not, and it's actually whether there's many single mutants being produced each generation or not. If n times mu is large, the stochasticity of the single mutants doesn't matter. If it's small, it does. And just one thing I want to say is it got fairly different behaviors and parameter dependencies just by varying these population sizes, and they're kind of like different flavors of infinite population sizes. So this one up here, the really big population size, that was effectively infinite. We can go up higher and higher in population size. There's no more interesting regimes there. This one was, in the intermediate, was infinite from the point of view of the single mutants. We could totally ignore the stochasticity there, but not at all for the double mutants. It was very important that it was limited. And this one down here was infinite from the point of view of the single mutants, not in the sense that they were deterministic, they were stochastic, but in the sense that we could use this branching process approximation, which is actually only valid when there's no competition among the individuals, when the different individuals.
The single branching process approximation.
It was actually a multi-type branching process, but I didn't tell you.
No, I mean the single lineage approximation.
Yeah, exactly. Yeah. But even for a single lineage, right? Technically, it's not a branching process in a finite population, right? So there's lots of different scales.
I have a question. What is mu0 and mu1?
Oh, yeah, sorry. This is a leftover slide where I didn't assume symmetry between the loci. So you have, so that's why you have all these annoying numbers running around. Okay, so just all together, what I didn't tell you about were kind of the bad regimes where it was hard to adapt. So basically, you want to live in this area that we talked about to be able to find a complex adaptation easily. So that means you want at least a medium-sized population, not too small. I didn't say, but if you live down here, you're in that problem we talked about before, where you have to worry about wandering off in sequence space and going to the wrong place. Not diffusing over populations, over sequence space now, but the whole population wanders. And you need the single mutants not to be too bad. Just one, maybe for people who come from a population genetics background, the one surprising thing about this condition that the single mutants not be too bad is that normally we measure the selective coefficients relative to the inverse population size, relative to the strength of drift. That's not what we have here. It's relative to something that depends on the mutation rates and the selective coefficient of the complex adaptation. And actually, we can allow a bigger cost of the single mutants as we increase the population size, even though they're being selected against more efficiently. So you want to be careful with your intuition here.
That's because you get more of them.
Exactly. Lower frequencies, but more absolute number, and in this case, absolute number is all that matters. Okay, so yeah, what are some numbers here? I don't want to take these too seriously, but let's say we had two exact nucleotides that we wanted to change, point mutations, and if we change them both, we get a 1% advantage. Okay, well then we have a condition on the population size and the cost. Population size needs to be big enough, cost needs to be small enough so that we can see them. If we put in the mutation rate for E. coli, we get 1 million. That looks tiny compared to like the number that are living in your gut, but I want to be careful. I mean, because this is also a time scale, and 1 million generations of E. coli is a pretty long time. But if you use the full population size, it's less of a problem. RNA virus with a much higher mutation rate, get a much lower requirement on the population size. Something that's tiny for a virus should easily find all the double mutations that are beneficial, no problem. Okay, so that was all asexual. Let's mix in sex. I'm not going to go through this in detail. Just here's another one of these parameter slices. Where now we're looking at how the depth of the valley, so how bad the single mutants are, the strength of recombination, more recombination this way, how frequent sex is, and then how long it takes the population to adapt as a function of that. And then the height of this is just how long it takes in absolute terms, and I've colored it according to whether sex is helping or hurting the population. So two things kind of jump out at you. There's this really horrible mountain here where you can't adapt at all. That's where you're getting stuck because the mutations are individually bad and they're always recombining, so you're kept close to the product distribution, and every time you get a double mutant, it's split up and the constituent single mutants are selected out. And here, actually, the time to cross the valley is growing exponentially with population size. So basically, evolution's not going to cross this way. If it wants to cross that valley, it's going to find some trick to do it. And then the other main feature is we get this nice division right where we thought it should be, where the recombination rate and the selected coefficient roughly match. When recombination rate is higher than that, it tends over a lot of parameter space to break up those double mutants and hurt you. And when it's lower than that, it doesn't really break them up very much. So all it can do is help you by bringing together single mutants that were in the population. It's a little bit more complicated than that. That's not an absolutely perfect pattern, but you can see it's pretty roughly true. Okay, so you can just put this all together, and here's just a couple different parameter slices plotted together to show that we can change the parameters a bit, and you get very different dynamics, or even times across the valley. So we've got how long it takes to adapt over there as a function of population size for actually fairly small population sizes. For a mutation that provides a 5% advantage, and just you see this big split where for a recombination rate that's on the order of S but a little smaller, it gets very, very easy to adapt as the population gets big. For a recombination rate, it's an order of magnitude bigger, it becomes impossible. The points are simulations, the curves are analysis, and you could bring this. You could really bring these two recombination rates even closer to the selective advantage, and you would still see this huge jump at large populations. So we get totally different dynamics, just depending on some fairly minor quantitative changes in the recombination rate, and also then we get very different dynamics as a function of population size. Okay, so what does it all mean? Just again, the adaptations can spread no problem without any help from intermediate genotypes that can be as bad as you want or as absent as you want, as long as the advantage of the combination is bigger than the recombination rate among its constituent mutations, and you don't need a very big advantage for most organisms. You can get some limited amount of complex adaptation very easily as long as the population is large, the intermediate genotypes are not too deleterious, and it really helps actually if you have a little bit of recombination, not too much. And we can make all of these statements quantitative. And when you put in real numbers for them, there's no obvious reason at this level why you shouldn't have complex adaptation all the time. Actually, so the stuff I'm doing, well, I'm tagging along with Joanna on right now, it's very easy to write down a model where actually most adaptation is complex if you have a diverse population and the diversity doesn't hurt you too much. Okay, but so what do we need to do here? Actually, for some reason, I never really calculated the valley crossing times in detail for arbitrary complexity and recombination rates. I just started to do it during the break because I felt bad about that. And it looks like you can get, it looks like it's basically intuitive. If mutations are very hard to come by, if the mutation rate is really low, then you don't want to have, then sex is really bad because the breaking up the combination dominates. When mutations are fairly frequent, then it's really an issue of combining them, and then sex seems to help. But I just need to look at that in more detail. Really, to make this a more realistic model, we need to take the rest of the genome seriously. Right now, I've totally ignored it. I've assumed it's all static, or not doing anything interesting that interferes with the adaptation. And really, in most of these real populations, there should be all kinds of selection going on in the rest of the genome, and there can be all kinds of hitchhiking of these individual constituent mutations on adaptation that's going elsewhere in the genome. There can be interference. So we need to know how that should actually work. But the most glaring problem right here is that there's no actual real fitness landscape in here. I'm just assuming one, no actual data. So really, the most important thing would be to actually take some natural population, maybe cancer, where we could hope to have some idea of what the actual fitness landscape looks like. And actually see some evolution in action, and see if this theory is helping us understand it at all. Thank you.
Questions for Daniel?
Nick, you're going to deny ever?
Could you test? No, so it's very neat way of putting together the theory, but how do we know it's happening? Could you test it by looking for amino acid substitutions that have come about by 2 or even 3 codon changes rather than 1? So the idea is it goes through a deleterious rare allele.
Yeah, I mean, just the hard part there is knowing that it really was deleterious in the middle.
The RNA secondary structures.
So I thought.
Photoshop has a paper on that, where he's got stem loop structures and that have had compensatory evolution at both sites. I mean, that one's got to be an example.
The problem is you need to convince people that it's not just purely compensatory. Okay, so the example here is it's one of these things where
it's
two.
It's not necessarily adaptive, but it's minimum compensatory.
Well, I really hear the dynamics. Really, it was crucially important that in this case where the two genes it was just 2 genes that needed to work together. They needed to fit together. That when they both changed, the new combination was actually better. If it's just the same, you get a different picture actually. Yeah. So you get a really clear valley there in the data, but it's hard to say, okay, this is really an adaptive change. Okay, thank you.